Okay, now we're going to go over the parasites of medical importance. And this is going to primarily be the protozoa and the parasitic worms, or the helminths. All right, parasitology. This is the study of eukaryotic parasites. So this is going to include protozoa. Remember, these are unicellular, animal-like eukaryotic organisms. And helminths, which are multicellular animals, or parasitic worms. And these guys cause about 1/5 of all infectious disease. Now, these organisms are going to be less prevalent in industrialized countries. We see them more in developing countries, and they're becoming increasingly common in AIDS patients. So first, let's talk about the major protozoan pathogens. Protozoa, remember, are going to be single-celled animal-like microbes. Most of them have some ability to move around, so most of them are motile. There's about 100,000 species, and only about 25 of those are going to be important pathogens for us. The life cycles of protozoa are going to vary species by species. However, most are going to propagate by simple asexual cell division of the active feeding cell, which is called the trophozoite. Many also undergo the formation of a dormant state, which is a cyst. And then others have a complex life cycle that can include asexual and sexual phases. Okay, so here's a table going over some parasitic protozoan pathogens, the main ones that we'll be talking about at least. And I've bolded the ones that you're likely to see a test question over. So this should be useful to you when you go back and study for the exam. In this lecture, we're going to talk about Entamoeba histolytica. This causes a chronic diarrhea. We'll talk about Naegleria fowleri and acanthamoeba. These are the brain-eating amoebas. We'll talk about Trichomonas vaginalis, the causative agent of trichomoniasis. This is an STD. We'll talk about Giardia and giardiasis, which is a condition of chronic diarrhea. We'll talk about Trypanosoma brucei, the causative agent of African sleeping sickness. We'll talk about Trypanosoma cruzi, the causative agent of Chagas disease. We'll go over Leishmania, the causative agent of leishmaniasis. We'll talk about Plasmodium. This is going to be a big one, guys. This is the causative agent of malaria. We'll go over Toxoplasma gondii, the causative agent of toxoplasmosis. Actually a very common subacute infection, but can become very serious in the immunocompromised and in the fetus. We'll talk a little bit about Cryptosporidium. This causes a gastroenteritis. Okay, first one to cover is Entamoeba histolytica. This guy is going to alternate between a large motile trophozoite, remember that's going to be the active feeding stage, and a smaller non-motile cyst. This is the dormant stage of the organism. We are going to be the primary hosts for it. Now what happens is you swallow the cysts, and they arrive in the small intestine. There's an alkaline pH, and digestive juices are going to stimulate those cysts to open up, and they release 4 trophozoites. Those trophozoites then attach to the small intestine, multiply, and then actively move about and feed. The amoeba can secrete enzymes that dissolve tissues. That's where it gets its name, histolytica, from histolysis, meaning tissue breakdown, okay? And these enzymes that dissolve tissues allow it to actively penetrate deeper layers of the mucosa, and this can leave ulcerations. However, in about 90% of patients, the infection is going to be asymptomatic. In those who do have symptoms, you're going to see dysentery, right? That's bloody diarrhea, abdominal pain, fever, and weight loss. And Entamoeba is carried by about 10% of the world's population. Okay, this is what the intestine looks like on gross pathology of a patient who has been suffering with amoebiasis or amoebic dysentery caused by Entamoeba histolytica. So you can see overall there is kind of a red appearance. It's erythematous. meaning that there was a lot of inflammation. And then you can see here there are ulcerations in the intestine. Again, from the ability of the organism to secrete those enzymes that break down tissues. Entamoeba histolytica can be life-threatening. Some life-threatening manifestations are hemorrhage, perforation of the intestine, appendicitis, and tumor-like growths called amoebomas. Okay, and these can cause intestinal obstruction. Entamoeba can invade the liver and it can invade the lung. And severe forms of amoebiasis have about a 10% fatality rate. Now, diagnosis is going to be made by microscopic examination of a fecal smear, and what they're looking for is the presence of those trophozoites or the cysts. Treatment, really what I want you to know about most of these protozoa and pathogens is treatment is going to be with metronidazole. All right, amoebic brain infections. These are going to be caused by Naegleria fowleri and acanthamoeba. And these guys ordinarily inhabit standing water, that is stagnant water. However, they have also been contracted using tap water to rinse sinuses. If you've ever seen or used a neti pot or something similar to that, you basically flush your sinuses with water. Make sure if you ever use that not to use tap water, use distilled or purified water instead. These guys can cause primary amoebic meningoencephalitis, and this is acquired through nasal contact with water or even traumatic eye damage. When it infects the brain, it is almost always fatal. There's only been 4 known survivors of this infection. So this is another pathology specimen. And what they've done is they've taken a section of the brain, so they took the brain and kind of sliced it like a loaf of bread. Now we're looking at one of those slices. And here you can see an area of hemorrhage and necrosis throughout the brain. And so imagine that just throughout the entire brain. This is a devastating disease. Okay, question. Entamoeba histolytica primarily invades. Well, if you go back and recall, we said that the cysts are going to be swallowed and arrive at the small intestine. Okay, so that's where they're going to attach, multiply, and move around and feed. So primarily they invade the small intestine. Remember, they can invade other sites like the liver and the lungs. Okay, now let's talk about Trichomonas. There's a few different Trichomonas species that are important to us. What they are is they're going to be small pear-shaped protozoa. They have 4 anterior flagella and an undulating membrane, so these guys are motile, and they're only going to exist in the trophozoite form. So these guys don't form cysts. Here you can see the pear-shaped organism, the anterior flagella here, and then here's the undulating membrane. Okay, so the one I want to talk about is Trichomonas vaginalis, and it causes an STD called trichomoniasis. The reservoir for Trichomonas vaginalis is the human urogenital tract. About 70% of those who are infected are asymptomatic. This guy is a strict parasite. It cannot survive long outside of us. And there's about 3 million cases yearly, so this is a very common STD. Symptoms in women are going to include foul-smelling green to yellow discharge, vulvitis, okay, so inflammation of the vulva, cervicitis, or inflammation of the cervix, urinary frequency, and pain when urinating. Symptoms in men are going to include urethritis, and remember this is inflammation of the urethra, a thin milky discharge, and occasionally prostatitis or prostate infection. Treatment is again going to be metronidazole. All right, Giardia intestinalis. This guy is also called Giardia lamblia. That's kind of an older name. You'll sometimes hear it called Giardia duodenalis as well. Okay, so all of these are the same organism. We typically just refer to it as Giardia. This guy is a pathogenic flagellate, meaning it has flagella. It has a unique symmetrical heart shape with this concave ventral surface that kind of acts like a suction cup. It has two nuclei, the trophozoite stage at least. And then the cysts are going to be small, compact, and multinucleate as well. Reservoirs for Giardia include a bunch of different mammals: beavers, cattle, coyotes, cats, and of course us. Cysts can survive for quite a long time outside of the body. They can survive for about 2 months in the environment, making them very problematic. And the condition that Giardia causes is called giardiasis. This is a diarrhea, abdominal pain, and flatulence. Okay, so I want you to know these symptoms for the test. It's also associated with greasy, foul-smelling stool. This is kind of just what the organism looks like in its trophozoite stage. You can see it has two nuclei, and then it has this ventral depression that kind of acts like a suction cup. Here is the cyst stage, and you can see it has multiple nuclei in it. Okay, so how is Giardia transmitted? It's going to be transmitted through the fecal-oral route. Okay, so this protozoan has been isolated from the intestines of a bunch of different mammals, but all mammals can potentially become infected with it. Both the trophozoites and the cysts are going to escape in the feces, but the cysts play the greater role in transmission, again, because they can last for a long time in the environment. Cysts are going to be ingested, usually with water or food, or they're swallowed after close contact with someone who's been infected, or close contact with a fomite. The infectious dose is pretty low, only 10 to 100 cysts required to cause infection. Now, prevention and treatment, there is a vaccine for animals against Giardia, but there's no human vaccine available. So our best way to prevent is going to be avoid drinking from freshwater sources, and of course, proper food handling, things like that. Treatment, again, you're going to see metronidazole. All right, now let's talk about the hemoflagellates. This is going to include species of Trypanosoma and Leishmania. Now, these guys are obligate parasites, okay, and they live in our blood and our tissues. They cause life-threatening and debilitating zoonoses. And remember, a zoonosis is an infection that can be spread between animals and humans. And now the way that these guys are spread is going to be through an insect vector, specifically blood-sucking insects that serve as intermediate hosts. Now, the hemoflagellates have complicated life cycles and undergo several morphological changes. They go from an amastogote to a promastogote to an epimastogote, then to a tripomastogote, okay? Don't worry too much about the details of this. Just know that they have a very complicated life cycle and can undergo different cellular forms. First, we'll talk about Trypanosoma. There's two species to talk about: Trypanosoma brucei, which causes African sleeping sickness, and Trypanosoma cruzi, which causes Chagas disease. Chagas disease is going to be endemic to Central and South America. Okay, Trypanosoma brucei, I want you to know, is the causative agent of African sleeping sickness. It's going to be spread by the bite of the tsetse fly. It's harbored by mammals, and when it infects us, it's going to cause a mild first stage of infection and then progress to a severe second neurological stage of infection. And this is where the effects of the organism on the central nervous system are seen. So neurological symptoms are going to include sleep disturbances, tremors, paralysis, and coma. And ultimately, if left untreated, this disease is going to result in death. There's two variants caused by two subspecies of Trypanosoma brucei. There's a Gambian strain and a Rhodesian strain, okay, and they're just in different parts of Africa. Now what happens is the tsetse fly has trypomastigotes in its saliva. So when it bites the human, it's going to inoculate the skin with those trypomastigotes. Those are then going to multiply in the blood and migrate to the spleen, the lymph nodes, and ultimately the brain, where they're going to cause damage. All right, and then there's Trypanosoma cruzi. This is the causative agent of Chagas disease. Trypanosoma cruzi is going to be harbored by mammals, including us, and it's going to be spread through the Reduviid bug. This guy is in Central and South America, as well as Mexico and some southern parts of the U.S. Now what happens is the Reduviid bug basically bites you and it defecates nearby. And when you go to scratch the bite, you get the bug feces in the bite and you inoculate yourself with Trypanosoma cruzi. So in Chagas disease, T. cruzi is going to multiply in muscle and blood cells. And those blood cells are then going to rupture and release large numbers of the trypanosomes into the bloodstream. There's an acute phase and a chronic phase. The acute phase, the symptoms are mild to severe fever, nausea, and fatigue. You can see a chagoma, which is a red, indurated, or hard swelling at the site of the bug bite. And if the bug bite is close to the eyes, you see a distinct condition called Ramagna's sign, which involves swelling of the eyelids. And this is because the person has inoculated their conjunctiva with Trypanosoma cruzi. Now, then the patient's going to enter a chronic phase, and the chronic phase is virtually asymptomatic. However, the trypanosomes are going to move throughout the body and interrupt the function of the heart, the brain, and the intestinal tract. And its effects on the heart can especially be devastating and lead to permanent heart damage, heart failure, arrhythmias, things like this that can actually lead to death. Okay, here's Ramagna's sign. There's unilateral eye swelling about 1 to 2 weeks after the Reduviid bug's bite. And this is going to be due to inoculation of the conjunctiva. Okay, so from rubbing the trypanosomes into the eyes. All right, Leishmania species. There's about 20 different species in this genus. And altogether, these guys are going to cause leishmaniasis. Leishmaniasis is zoonosis, and it's transmitted among mammalian hosts. And it's transmitted by female sandflies when they go to take a blood meal. Now, there's 3 ways that this disease manifests. There's cutaneous leishmaniasis. This tends to be caused by L. tropica. There's mucocutaneous leishmaniasis, or Espundia. This tends to be caused by L. brasiliensis. And then there's visceral leishmaniasis, or Kala-Azar. And this tends to be caused by L. donovani. So how is leishmaniasis transmitted? Well, it's going to be transmitted through the bite of the female sandfly. She's going to inject the infective stage of the protozoa when she goes to take a blood meal. Now, only the females take blood meals. They need the blood in order to develop their eggs. Now, after the infective stage of the protozoa is injected, the protozoa are then going to be phagocytosed by macrophages. They're going to multiply in those macrophages and then break out of them to infect more cells. This disease is endemic to equatorial regions that provide favorable conditions for the sandfly. And so the people who are going to be at risk for this disease, at least the people that we tend to see, are going to be travelers and immigrants. Culture and diagnosis. Diagnosis is going to be through microscopic visualization. And then there's no vaccine, and the only way really to prevent leishmaniasis is to avoid the female sandfly. All right, so cutaneous leishmaniasis, sometimes this is also called an Aleppo boil, and what it is it results in a localized ulcerated sore. Then you've got Espundia, which is also mucocutaneous leishmaniasis. This is going to cause a skin and mucous membrane infection of the head, okay? And this tends to be a chronic infection. And then we have systemic or visceral leishmaniasis, also called Kala-Azar. With this, you get a high intermittent fever, weight loss, anemia, enlarged spleen, enlarged liver, and enlarged lymph nodes. And what happens is the parasites invade the spleen, the liver, and the bone marrow. And if left untreated, this condition has a 95% mortality rate. In fact, leishmania is the second biggest protozoan killer, the first being Plasmodium, which we'll talk about here shortly. So these are some examples of the ulcerative lesions that occur in cutaneous leishmaniasis. Okay, we have a question here. Hemoflagellates, and remember that's going to be Leishmania species and the trypanosomes. Hemoflagellates are transmitted by what? Okay, so recall that the trypanosomes, T. brucei, is going to be transmitted through the tsetse fly. T. krusei is going to be transmitted through the reduviid bug. And leishmania is going to be transmitted through the female sandfly. So all of these are transmitted through insect vectors. All right, the apicomplexan parasites. These are protozoa. They're also called sporozoans. This is an older term for them. They all lack locomotor organelles, so they don't have flagella in the trophozoite state. They're going to alternate between sexual and asexual phases and between different animal hosts. Sometimes they're going to utilize entirely different hosts for their sexual and asexual stages. Okay, so these guys have kind of complicated life cycles. Most of them form specialized infective bodies called sporozoites that are transmitted through arthropod vectors, contaminated food and water, or by other means. And the three that we're going to talk about are going to be Plasmodium, Toxoplasma, and Cryptosporidium. Okay, Plasmodium. This is the agent of malaria. Plasmodium is an obligate intracellular parasite, and there's 5 species that cause malaria, okay? So Plasmodium malariae, Plasmodium vivax, Plasmodium falciparum, Plasmodium ovale, and Plasmodium knowlesi. The vector here is going to be the female Anopheles mosquito. And I want you to know that the female Anopheles mosquito feeds at night. Another way that you can get malaria is through a blood transfusion, and it can also pass through the placenta, so from mother to fetus. Malaria is a significant global disease with 200 million new cases every year and around 435,000 deaths every year. And you can see it's going to be concentrated kind of along the equator here. Okay, the Plasmodium life cycle. So it's a little bit complex, and I'm going to go through it with you. But it's going to involve an asexual phase that occurs in us and a sexual phase that occurs in the mosquito. Now, what happens is the female mosquito takes a blood meal from us. And when she does that, she injects sporozoites into our bloodstream, which are then going to travel to the liver. Now, these sporozoites are asexual, and when they get to the liver, they're going to undergo asexual division. And when they do, they produce merozoites. Now, the liver cells are going to swell and burst from the accumulation of these merozoites, and that's going to release thousands of them into the blood. Now, those merozoites are going to go attach to and enter the red blood cells. When they get into the red blood cells, they're going to convert to trophozoites. Remember, this is the active feeding form of the organism. And these trophozoites are going to feed on our hemoglobin and multiply. The trophozoites mature into schizonts, which are multinucleated asexual forms of the parasite. And these schizonts contain many, many, many merozoites. Some of these merozoites are going to differentiate into gametes. Now the schizont grows until the red blood cell bursts, and this is going to release tons of merozoites into the bloodstream that go on to infect more red blood cells. So this cycle, the merozoite to trophozoite to schizont to merozoite cycle, is going to repeat every 48 to 72 hours, and this is what causes the cyclical fevers we see in malaria. Now remember, some of those merozoites go on to differentiate into gametes. So the next phase is the sexual phase of Plasmodium, and it's going to occur in the mosquito. So the female Anopheles mosquito comes, she takes a blood meal, and when she does that, she's going to draw infected blood containing those gametes. And then the male and the female gametes are going to fertilize in that mosquito's gut, forming a zygote, which develops into thousands of sporozoites. Those sporozoites migrate to the salivary glands of the mosquito. And so on her next blood meal, she can inject sporozoites into our bloodstream. And remember, sporozoites are going to be the infective form of the parasite. Now note here that the mosquito is not only the vector in this disease, but is also the definitive host, because in the definitive host is the one in which the parasite undergoes sexual reproduction. Okay, so let's go over it. So you have a female Anopheles mosquito. She is infected with Plasmodium, and she has Plasmodium sporozoites in her salivary glands. She comes to us, she bites us, and draws in a blood meal. When she does that, she injects sporozoites into our bloodstream. Now those sporozoites travel to the liver where they infect liver cells. You then enter the liver stage of the infection. Now the parasite reproduces asexually in those liver cells, that bursting the cell and releasing merozoites into the blood. These merozoites go on to infect red blood cells. Now we're at the red blood cell stage. The parasites reproduce asexually again, remember, because this is in us, so we're the asexual stage. They reproduce asexually in the red blood cells, bursting those red blood cells open and causing cycles of fever. And then, of course, releasing those merozoites into the bloodstream where they can go on to infect more red blood cells, okay? And this can just continue and continue and continue. Now remember, some merozoites are going to differentiate into gametes. And when the mosquito draws her next blood meal, she's going to take some of those gametes up with it. The gametes are going to fertilize in the mosquito's intestine, and then they're going to develop into sporozoites, which then infect the mosquito's salivary glands. Then when she takes a blood meal, she can inject us with those sporozoites, and the whole cycle starts over again. One thing I want to point out here is that during the liver stage, the parasite can actually remain in the liver cells for a long time, years in some species. So this liver stage can cause recurrence of malaria, even sometime after the malaria has, the symptoms have dissipated. Okay, so this can lead to latent infection. And it's one reason why if you've ever had malaria, you are not allowed to donate blood because of the potential for latent infection. And now recurrence can occur even after treatment because our drugs that we use to treat malaria tend to target the red blood cell stage. And they don't target the parasite when it's in the liver stage. So this is why the parasite can hang around and cause recurrent disease years later. Okay, so there's about a 10 to 16 day incubation period for malaria, and the first symptoms are going to be malaise, fatigue, aches, and nausea, followed by chills, fever, and sweating. And these symptoms are going to occur at different intervals depending on the species, but generally at 48 to 72-hour intervals. And this is because those red blood cells all tend to swell and rupture at the same time. We said the interval, the length, and the regularity of symptoms are going to reflect the type of malaria. You've got P. vivax and ovale, which cause fever every 2 days. P. malariae, which causes fever every 3 days. And then you've got Plasmodium falciparum, which causes the most severe form of malaria. And Plasmodium falciparum tends to have a persistent fever. It can also have a cough and weakness, severe weakness. Complications of malaria include hemolytic anemia. So you're going to have a decrease in circulating red blood cells because those red blood cells are being destroyed, right, when the parasite bursts out of them. Then you're going to get some organ enlargement, especially of the spleen and the liver. And this is because of that cellular debris that's circulating. Remember, the red blood cells are being broken down, so are the liver cells. So debris from those broken down cells are traveling through the bloodstream and get caught up in the spleen, the liver, and then finally, the most severe complication is cerebral malaria. And what happens in cerebral malaria is that the infected red blood cells block the small blood vessels of the brain. And this is going to cause a lack of oxygen to the brain. So cerebral malaria is one of the most serious complications, primarily of falciparum malaria. What happens here is the small blood vessels in the brain are going to become blocked because of those infected red blood cells. And so what happens is you don't get good blood flow through those vessels, so you don't get oxygen to the brain tissue. And so a decrease in oxygen in the brain can cause coma and death. The blood-brain barrier can also lose its integrity due to severe inflammation, and this will let in things like cytokines and bacteria and the parasites themselves, and this can damage the brain, and the brain swells. Now, in general, malaria has the highest death rate in the acute phase, especially in children. Malaria caused by Plasmodium vivax and Plasmodium ovale are going to be subject to relapses because, like I said, some infected liver cells can harbor dormant protozoans for years. Diagnosing malaria. So what they do is they look at a blood smear, and if you see the trophozoite inside of red blood cells in the blood smear, that indicates the presence of Plasmodium. You can also diagnose based on symptoms, so recurring chills, fever, and sweating. There are also some rapid malaria tests that you can utilize that are looking for antigens. Treatment. There are several different drugs that can be used to treat malaria, but the ones I want you to know about are going to be the ones based on quinine drugs. So things like chloroquine and primaquine. Malarial control. So a big portion of malaria control is going to be mosquito abatement. So the idea here is to drain water. So you're reducing breeding sites when you do that. Apply insecticide to kill the mosquitoes. On your own body, you can use insect repellents. Sleeping under mosquito nets is useful, again, because remember the mosquito, the female Anopheles mosquito, feeds at night. So you're at highest risk of getting malaria at nighttime. So sleeping under a net that keeps the mosquitoes out is a good preventive strategy. And then, of course, human chemoprophylaxis. This is taking anti-malarial medications ahead of time so that if and when you do get infected with Plasmodium, you can control the infection right away. Now, vaccination for malaria has been in development for decades. And one of the reasons why it's been so difficult to develop is because there are so many life stages to target in the Plasmodium life cycle. The first vaccine for malaria was released just 4 years ago. This is the RTS-S vaccine, and it targets Plasmodium falciparum to prevent liver infection in children under 5 years of age. So this vaccine is primarily targeting the sporozoites of Plasmodium falciparum. Here is an example of a malaria net. And if you've ever traveled to a country where malaria is present, you have probably slept underneath one of these. Now, a lot of people don't know, but malaria used to be endemic to the United States. During the 1600s to the 1800s, it was widespread in many parts of the U.S., especially where the Anopheles mosquito thrives, so generally in the South, the Southeast. But cases would occur as far north as New York and Michigan during the warmer months. Even American Civil War soldiers suffered from malaria. Now, starting in the 1940s, the U.S. launched a massive public health campaign. They drained swamps, they improved sanitation, sprayed DDT and other vector control, used other vector control methods, screened houses, improved healthcare, and we started treating infected people. By 1951, malaria was considered eliminated from the United States. However, it still can occur as an imported disease. Travelers returning from malaria-endemic countries can bring it back, and if they get bitten by an Anopheles mosquito here, that Anopheles mosquito can spread it to another person. So there is a small chance for local transmission, and this has happened occasionally. There have been small outbreaks in Florida, Texas, and Maryland as recently as just 2 years ago. Okay, Toxoplasma gondii and toxoplasmosis. Toxoplasma is an intracellular epicomplexan parasite with extensive distribution throughout the world. It lives naturally in cats. Cats are actually the definitive host for this parasite, right? So that's where the parasite reproduces sexually. And the cats harbor the cysts of this parasite in the GI tract. So how do you get toxoplasmosis? Well, you acquire it either by ingesting raw or undercooked meats, because toxoplasma can form cysts in the muscle of infected animals, or you can acquire it through substances that have been contaminated by cat feces, because those cysts reside in the GI tract of cats. Now, most cases of toxoplasmosis are going to be subacute. That means they go unnoticed. However, in the fetus and in AIDS patients, severe complications can occur, including brain and heart damage. Now, the treatment for toxoplasmosis is going to be sulfonamides. All right, so here's an overview of the life cycle of toxoplasma. The cat is the definitive host, and remember, that's where the sexual phase of the organism is going to take place, in the definitive host. Now, the infected cat is going to shed oocysts in its feces, and these oocysts can contaminate food or water, and then we ingest the contaminated food or water, or animals do, and that's how we get infected. Now, in an animal, when they are infected, the organism then forms cysts in their muscles. Now, if you ingest that meat, raw or undercooked, you can become infected with toxoplasma. In fact, this is how cats become infected. So you have cat 1 who is infected, who sheds the oocysts. The oocysts contaminate a food source or a water source, and a mouse or a rat comes along and gets infected. Now, one of the side effects of toxoplasmosis in rodents is that they lose the fear, the natural fear that they have of cats. So you'll see an infected mouse or an infected rat walk right up to a cat. And so cat number 2 sees this mouse or rat and ingests it, not knowing that it is infected. And so cat number 2 becomes infected with toxoplasma. And the cycle goes on and on. Now, we can get infected through either ingesting the oocysts or through ingesting the tissue cysts. And in us, it tends to be a subacute infection as adults. However, it can cross the placenta. And when it does that, it can have devastating effects on the fetus. In fact, this is one of the reasons that they tell pregnant women not to do the cat litter box if they have cats at home, is to avoid potentially contaminating themselves with Toxoplasma oocysts. Now, this is a pretty common infection. Like I said, in most people, it's going to be pretty much asymptomatic, maybe a low-grade fever, some swollen lymph nodes. It's estimated that 1/3 of the world's population, including about 11% of the U.S., is infected with Toxoplasma gondii. Again, this infection is not usually much of a problem unless you are a fetus or unless you are immunocompromised or become immunocompromised while chronically infected with Toxoplasma. All right, Cryptosporidium. This pathogen causes cryptosporidiosis, which mimics gastroenteritis. For the exam, I want you to know the symptoms of cryptosporidiosis. That's going to be headache, sweating, vomiting, abdominal cramps, and diarrhea. And I want you to know that AIDS patients can suffer a chronic, persistent form of this disease. All right, I want you to be familiar with the Cryptosporidium life cycle. So what happens is the oocysts are going to be ingested by us with contaminated water or food, and it's been contaminated by feces from infected animals. That oocysts then open up once they reach the intestine, and they release sporozoites that attach to the epithelium of the small intestine. Cryptosporidium then penetrates the intestinal cells and lives in them intracellularly, just below the plasma membrane. So these are intracellular pathogens, but they're considered extracytoplasmic, and I'll show you a picture of that here in a minute. They undergo asexual and sexual reproduction in these intestinal cells. This means that they're monoxenous, and all that means is that's a parasite that uses a single host for its asexual and sexual life stages. So they undergo asexual and sexual reproduction, producing more oocysts, which are then going to be released into the gut and then excreted in the feces. And then they can go on to infect other humans or animals if they're ingested. And a big problem with Cryptosporidium is that the oocysts are resistant to disinfection with chlorine. So this causes problems for water sanitation efforts. Okay, so this is what Cryptosporidium looks like under the microscope. Down here you can see kind of a zoomed in diagram. So you can see the parasite here underneath the plasma membrane of the enterocyte. But you can tell it's not in the cytoplasm of the intestinal cell. And so they kind of look like little blebs coming off of the intestinal cells here. All right, so Babesia was the first protozoan found to cause a disease. It was found in 1893 to cause Texas cattle fever, or bovine babesiosis. And this was caused by the species Babesia bigemina. It was also discovered that it was transmitted by a tick. So this is the first protozoan found to be associated with a vector. In us, it causes human babesiosis, which is a relatively rare zoonosis. This is associated with infected rodents, and the infection is going to resemble malaria. All right, so here's the life cycle of Babesia. So you can see it kind of goes back and forth between the white-footed mouse and the Ixodes tick. And the white-footed mouse is going to involve the asexual phase of their life cycle, while the tick is going to involve the sexual phase of the life cycle. So that means the tick is the vector as well as the definitive host. Now, sometimes the tick goes and it bites us, and we kind of become dead-end hosts. Okay, there's really nowhere for this organism to go once it has infected us. It can theoretically be spread human to human by ticks. However, that is incredibly rare. More likely for human-to-human transmission would be through blood transfusion. So this is one thing that donated blood is screened for. All right, question. What makes cryptosporidium particularly challenging to control in water supplies? Well, if you go back and recall that the oocysts are going to be resistant to disinfection with chlorine. Which cell type does the Plasmodium merozoite invade? So let's go back to the life cycle of Plasmodium. And kind of what I want you to take away from this big, complicated life cycle is I want you to know that the sporozoites are the infective stage. These are going to be the guys that infect the liver cells. And that the merozoites are going to be the stage that infects red blood cells. All right, now let's talk about helminths. Helminths are parasitic worms. The adults are large, multicellular animals with specialized tissues and organs. Adult worms mate and produce fertilized eggs that hatch into larvae. And then the larvae then mature in several stages to adults. The sexes can be separate or hermaphroditic. It just depends on the species. Remember that adulthood and mating is going to occur in the definitive host. Larval development is going to occur in the intermediate host. And then a transport host is a host that experiences no parasitic development. And there's 4 basic patterns of life and transmission. All right, so here are the 4 basic helminth life and transmission cycles. We have cycle A, cycle B, cycle C, and cycle D. Cycle A is going to involve direct fecal-oral transmission. So what happens here is humans ingest the eggs. The worms then mature in the intestine. And the human sheds the eggs in the feces, and the cycle can start over again. Examples here would be Ascaris or Trichuris. Okay, in cycle B, transmission is going to be through larval penetration of the skin. What happens is eggs hatch in the environment into larvae. The larvae penetrate the skin of the host. They migrate to the intestine, where they mature and they mate, and they produce eggs. Those eggs are going to be shed with the feces. They're going to end up in the environment where they hatch into larvae. The larvae develop, and then they're ready to penetrate the skin of the next host. The example here would be hookworms. All right, cycle C. Cycle C is going to require an intermediate host. What happens here is humans are going to eat meat that has cysts in it. Those cysts are going to release larvae. The worms make it to the intestine where they mature. They mate, and the human sheds eggs in feces. The eggs then go on to infect an intermediate host, and they insist in the muscle of that intermediate host. We eat the muscle, right? We eat meat. If we eat undercooked meat that has been infected with these cysts, the cysts are going to release larvae, and the cycle continues all over again. Now, one interesting thing about cycle C is that the eggs can go on to infect an intermediate host, or they can infect a human. And when the human ingests eggs rather than the cysts in meat, ingesting eggs is going to cause another variation of the disease, another manifestation. And what's going to happen is that the organism is going to insist in our muscles, in our tissues. The example here is going to be tinea, and we'll talk about this in more detail when we talk about cysticercosis. All right, in cycle D, what's going to happen here is humans are going to eat larvae either in meat, or the larvae are going to penetrate the skin. Worms are going to mature in the intestine or in the bladder. The humans are going to shed eggs, either in their feces or in their urine. Eggs are going to hatch into larvae in water. Then there's going to be a second larval stage that's going to involve an intermediate host, usually or often a snail. And then there's going to be a second larval stage, and that larvae can go on to infect humans through ingestion, right, through eating infected animal meat, or through penetration of the skin. And we're going to see this with the flukes. Okay, this is a table going over the major helminthic pathogens that we are going to talk about. So first, we'll go over the nematodes. These are also called the roundworms. And we'll talk about intestinal nematodes. So you're going to be agents like Ascaris lumbricoides or Enterobius vermicularis. And then we'll talk about pathogens such as the tissue nematodes. Then we'll talk about the trematodes. These are also called flukes. And primarily we'll talk about schistosoma and Opisthochus. Then we'll talk about the cestodes. These are also called tapeworms. We'll talk about the beef tapeworm and the pork tapeworm. I won't talk about the fish tapeworm, but one thing I want you to notice is that they're all going to be acquired through consumption of undercooked meat. Okay, so for the beef tapeworm, it's going to be undercooked or raw beef. Pork tapeworm, it's going to be pork. And for the fish tapeworm, it's going to be fish. All right, pathology in helminth infections is going to arise from the effects of worms feeding on and migrating through the body's tissues, through the actual physical accumulation of worms. They can cause intestinal obstruction, for example, and through the accumulation of worm products. Diagnosis can be a combination of things. So a blood cell count will usually show eosinophilia during helminth infections. Some serological tests are available. A big way to diagnose is to look for eggs, larvae, or even adult worms in the feces. But you can also examine other samples, such as sputum, urine, blood, or tissue. Now, several anti-helminthic medications exist. And recall that helminths are going to be eukaryotic, so they are more similar to us than, say, bacteria or a prokaryotic pathogen would be. So because they're more like us, their cellular physiology resembles ours. So finding drugs that target helminths, specifically helminths, while leaving our cells alone can be challenging. Some anti-helminthic drugs are going to suppress a metabolic process that's more important to the worm than it is to us. And then other anti-helminthic drugs are going to inhibit the worm's movement, basically paralyze the worm, preventing it from being able to maintain its position in a certain organ and complete its life cycle. All right, and some helminths have started to develop resistance to these drugs. Now, in some cases, surgery may be required to physically remove the worms. For example, remove them from the appendix, from an intestine, or from the bile duct. All right, first let's talk about the nematodes. These are also known as roundworms. So the vast majority of these guys are going to be free-living soil and freshwater worms, but about 200 of them are parasitic, And there's 50 species that specifically affect humans. These guys are elongated, cylindrical worms with protective cuticles, circular muscles, a complete digestive tract, and separate sexes. The human parasites are going to be divided into intestinal nematodes and tissue nematodes. Now, intestinal nematodes are going to develop to some degree in the intestine. And this is going to include pathogens such as Ascaris, Strongyloides, and the hookworms. Tissue nematodes, on the other hand, are going to spend their larval and adult phases in various soft tissues other than the intestine. For example, the filarial worms, some of which spend their time in the lymphatic system, some of which can infect the eye. All right, let's start with Ascaris lumbricoides. This is the causative agent of ascariasis. Ascaris lumbricoides is a large intestinal roundworm. It is the most common worm parasite found in humans. Most cases in the U.S. are going to occur in the southeastern states. Ascaris spends its larval and adult stages in us. It releases embryonic eggs in the feces, and then those eggs are spread to other humans and can contaminate food, drink, or other objects. Adult worms are going to complete their cycle in the intestines and reproduce there and produce about 200,000 eggs a day. So what does Ascaris do when it infects us? Well, it's going to enter the body as eggs, and those eggs are going to hatch into the larval stage in the intestine. From there, the larvae are going to migrate to the circulatory or the lymphatic system. Okay, so they're going to get into the blood or the lymph, and from there make their way to the lungs. They get into the lungs, and they're going to be coughed up. So they're going to migrate up the respiratory tree. The patient will cough the worms up, and then swallow them down. The larvae are going to get into the stomach and then down into the intestinal tract, where they then mature into adult worms, and they're going to take up residence in the intestines. There they will mate, and the adult female will lay eggs in the intestine, which then get passed along with the feces. Symptoms are going to be primarily digestive tract symptoms, okay? You can also see inflammatory reactions along worm migratory routes. A big giveaway that the patient might have a worm infection is going to be a high eosinophil count, and you might see pneumonia during the lung stage of the worms. Okay, so I have a few pictures here. On the left is an x-ray, and this is a barium swallow. So what they do is they give the patient this stuff called barium, and it shows up white on the x-ray. So the patient swallows this barium, and it highlights their digestive tract. So here's the stomach, here's the duodenum, and then here we're getting into the rest of the small intestine. And you can see these markings here kind of look thread-like. Those are worms in the small intestine, packing the small intestine. Over here you can see plain film x-rays. And you see in this child distended bowel loops with air fluid levels. And what's happening is right here around the cecum, there is an obstruction caused by the worms. So what they did was they took this patient to the OR, opened them up, and then opened up their intestine and physically removed the worms that were causing the obstruction. Okay, Trichuris trichiura. This is the whipworm. We are its sole host. Trichuriasis has its highest incidence in the tropics. What happens is the eggs are going to hatch in the intestines. The larvae are going to attach to the intestines and penetrate the outer wall and develop into adults there. Females are going to lay their eggs, about 3,000 to 5,000 eggs a day. And the worms can go on to pierce capillaries, causing localized hemorrhage. Heavy infestations can cause malnutrition, dysentery, and rectal prolapse. All right, this is a colonoscopy of a patient with a whipworm infection, and you can see the worms lining the colon here. And what they've done is they've attached to the wall of the colon. All right, Enterobius vermicularis. This is the pinworm, sometimes also called the seat worm. This is going to be the most common worm disease of children in temperate zones, so that includes us in the U.S. Transmission is the cycle A type. So what happens is the freshly deposited eggs are sticky, so it's really easy for them to lodge beneath fingernails or lodge onto fomites. When they dry, the eggs can even become airborne and settle in house dust. So what happens is the eggs are ingested from contaminated food or drink or through self-inoculation. So if the patient gets eggs on their fingers and then puts their fingers in their mouth, eggs are going to hatch in the small intestine and there release larvae that then migrate to the large intestine. In the large intestine, the larvae are going to mature into adult worms and mate. Then what happens is the female moves to the anus at night and lays her eggs just outside of the anus, and this causes intense perianal itching. And so the patient, usually a child, will reach down there and scratch and unfortunately get the eggs on the fingers. And again, the cycle can repeat. The eggs can get put on fomites, or the patient can self-inoculate themselves again, put their fingers in their mouth and take in the eggs. This is a big problem with children who are not very hygienic. To diagnose it, you use the tape test. So what they do is they take a little piece of Scotch tape, you press it against the perianal skin, and then you take that Scotch tape and you press it against a microscope slide. So what you're trying to do is you're just trying to pick up the eggs, get it onto the Scotch tape, and then transfer the eggs over to a microscope slide. And then you can look at it underneath the microscope and identify the eggs. All right, pay attention to this question because you're going to see a very similar question on the test. Ascaris lumbricoides, A, spreads to human by ingestion of Ascaris eggs in food. B, is an intestinal roundworm. C, larvae penetrate into lymphatics and capillaries around intestines. D, larvae migrate to the pharynx, get swallowed, and return to intestines to mature. Or E, all of the choices are correct. Well, if you recall, Ascaris, we do get Ascaris infections by ingesting eggs. Ascaris is an intestinal roundworm. The Ascaris larvae are going to penetrate into lymphatics and capillaries, and from there migrate to the lungs. From the lungs, they're going to migrate up the throat, get swallowed, and return to the intestines to mature. So all of these choices are correct. Okay, Enterobius vermicularis is A, an intestinal worm that easily contaminates fingers and fomites. That's true. B, common only to the tropics and subtropics. That's incorrect. Remember, this is going to be a temperate zone. Well, it's common globally, but it's the most common worm infection in temperate zones. C, often fatal in heavy infestations. That is incorrect. D, a whipworm, also incorrect. So the correct answer here is A. Hookworms are so named because they have characteristic curved ends and hooked mouths. The species to be familiar with are Necator americanus and Ankylostoma duodenale. Humans are going to shed the eggs in feces, which then are going to hatch into larvae and burrow into the skin of bare feet. From there, the larvae are going to travel from the blood to the lungs, proceed up the bronchi and throat, and again are going to be swallowed down into the stomach. There, in the intestine, the worms are going to mature and reproduce in the small intestine and complete their life cycle. We are the definitive host. Hookworms can cause pneumonia, nausea, vomiting, cramps, and bloody diarrhea. And blood loss can be significant, resulting in anemia. All right, Strongyloides stercoralis and strongyloidiasis. Strongyloides is the threadworm. These are tiny roundworms that complete their life cycle in humans or in the moist soil. What happens is the larvae penetrate the skin, and then again, you're going to see this over and over, migrate to the lungs, are coughed up and swallowed, and then complete development in the intestine. Heavy worm loads can cause pneumonitis or inflammation of the lungs, eosinophilia. Remember that. You're going to see eosinophilia in worm infections. It can also cause bloody diarrhea, liver enlargement, bowel obstruction, and malabsorption. Trichinella spiralis. This is the causative agent of trichinellosis. You acquire trichinella from eating undercooked pork or even bear meat. You'll see that this is more common in Alaska, where bear and seal meat are consumed. The larvae are going to migrate from the intestine to the blood vessels, the muscle, the heart, and the brain, where it's going to form cysts. The first symptoms are going to be flu-like symptoms, maybe a little bit of diarrhea as well. And then second symptoms are going to come in. You're going to see muscle and joint pain, shortness of breath, and pronounced eosinophilia. Now, there's no cure after the larvae have encysted. All right, tissue nematodes. So we talked about the intestinal nematodes that at least complete part of their life cycle in the intestines. The tissue nematodes are going to complete their life cycle in human blood, lymphatics, or skin. These are going to be the filarial worms. These guys are elongate, they have filamentous bodies, and they're going to be spread by biting arthropods. They are the causative agent of filariasis, which is a chronic deforming disease. The species we're going to talk about are going to be Wuchereria bancrofti. This is the causative agent of elephantiasis. Onchocerca volvulus, the causative agent of river blindness. And Loa loa, the eye worm. Okay, Wuchereria bancrofti causes Bancroftian filariasis. This is a tropical infection spread by mosquitoes. The mosquito is going to deposit the larvae, which are going to then move into the lymphatics and develop into adults. Chronic infection is going to cause blockage of the lymphatic circulation, which is going to cause lymphedema, a huge amount of lymphedema. And this condition is called elephantiasis. You see massive swelling in the extremities and sometimes in men in the scrotum. All right, so this is what elephantiasis looks like. You can see massive swelling in the lower extremity here. And again, this is due to the lymphatics being blocked. So this lymphatic fluid cannot drain from the limb. Okay, Onchocerca volvulus is the causative agent of river blindness. This pathogen is going to be transmitted by biting black flies. The larvae are going to develop into adults in the subcutaneous tissues. From there, the adult females are going to migrate via the blood to the eyes, and this is going to provoke an inflammatory reaction. Now, with nematodes that are also co-infected with Wolbachia bacteria, you're going to see a huge inflammatory response in the eye, and this is going to damage the cornea, and this is what causes river blindness. Now, eliminating the Wolbachia bacteria from the nematode is generally going to result in the nematode either dying or becoming sterile. Then there's Loa loa. This is the African eyeworm. It's going to be spread by the bite of small flies. This guy is temperature sensitive, so it's going to hang out just under the skin, and it can enter the eye. Treatment, you can give medication, but you can also pull the worm from a small hole that's been punctured in the conjunctiva. Here you can see the eye worm just underneath the surface of the eye. All right, moving on, let's talk about the trematodes. These guys are also called flukes. These guys are flatworms with ovoid leaf-like bodies. You can see that kind of down here, has sort of a leaf-like body. They have fully formed digestive, excretory, neuromuscular, and reproductive systems. However, they're going to lack a circulatory and respiratory system. Animals such as snails or fish are usually going to be the intermediate hosts, while we are the definitive hosts for this organism. Okay, so these guys are primarily going to go through cycle D. And what you're going to see over and over again is you're going to see that the larvae are going to penetrate through our skin. They're going to mature inside of us and produce eggs. We are going to shed the eggs to the environment, and then those eggs are going to hatch into larvae and complete part of their life cycle in an intermediate host, usually a snail, okay? Okay, so let's talk about the blood flukes first. These are the schistosomes. They cause schistosomiasis. The causative agents here are Schistosoma mansoni, Schistosoma japonicum, and Schistosoma hematobium. So adult flukes are going to live in humans, and they're going to produce eggs, and those eggs are going to be shed into water. The eggs hatch into an early larval form, and this early larval form is going to develop in the freshwater snail into a second larval form, the cercaria. Now it's the cercaria that penetrates our skin. They're going to migrate to the liver and mature there. Then the adults migrate to the intestine or to the bladder, depending on the species, and shed eggs. And then those eggs are going to be released with either the feces or the urine into the environment. And the cycle can start all over again. Schistosomiasis, when it takes up residence in the bladder, is going to be a huge risk factor for bladder cancer. All right, then we have the liver flukes. This is going to be Opisthorchis sinensis and Fasciola hepatica. Opisthorchis cycles between mammals, snails, and fish. Now you're going to see this over and over again with the flukes. Snails tend to be an intermediate host. With Opisthorchis, humans are going to be infected by eating inadequately cooked fish, and the fluke is going to migrate to the biliary tree. With Fasciola hepatica, this guy is going to cycle between herbivores, snails, and aquatic plants. We are infected by eating raw aquatic plants, and the fluke is going to lodge in the liver. All right, and then there's the lung flukes. This would include Paragonomus westermanni. This guy cycles between carnivorous animals, snails, and crustaceans. We get infected by eating undercooked crustaceans. Worms are going to migrate from the intestines to the lungs. All right, question. The organism that causes elephantiasis is multicellular with a complete digestive tract and reproductive organs. Well, if you recall, the organism that causes elephantiasis is Wuchereria bancrofti, and it's a worm. It's a filarial worm, so it's going to be multicellular, and it does have a complete digestive tract and reproductive organs like other worms do. So the answer here is true. Question, which of the following is an intermediate host for a trematode? This is very likely to be a question you will see on the test. And if all of the pictures I gave you don't give it away, it is the snail. All right, and then finally, let's talk about the cestodes. These are the tapeworms, also called flatworms. These guys have long, thin, ribbon-like bodies that are composed of these sacs called proglottids and a head that's called a scolex. And the job of the scolex is just to grip the intestine, okay? It doesn't actually have like a mouth or a digestive tract there. Each proglottid is going to be an independent unit that's adapted to absorbing food and making and releasing eggs. These tapeworms are going to be the principal causes of human teniasis. We're going to talk about Tenia saginata and Tenia solium. All right, so a little bit of tapeworm anatomy. Here you have the scolex. This is the part of the worm that's going to attach to the intestine. Here you have the body, and you can see immature proglottids here near the scolex, and then as you move down the body, the proglottids become mature. And inside of each proglottid, there are testes, there are ovaries. So these worms are hermaphroditic. So each proglottid is essentially just an egg-making unit. And remember, tapeworms do not have a mouth and they don't have a digestive system. Instead, the proglottids are going to absorb the pre-digested nutrients from the host's gut. All right, first we have Tinea saginata. This is the beef tapeworm. It's very large. It can have up to 2,000 proglottids, and we are the definitive host. Here is a close-up image of the scolex of Tinea solium, displaying 4 suckers and 2 rows of hooklets. So this is the attachment apparatus of the worm. So animals are going to become infected by grazing on land that's been contaminated with human feces. We are going to become infected by eating raw beef or undercooked beef in which the larval form has encysted. In us, the larvae attaches to the small intestine, and there it becomes an adult. This guy is going to cause very few symptoms, vague abdominal pain and nausea, and you're going to see proglottids in the stool, but other than that, not a huge amount of symptoms with this infection. So here you have an infected human. They're releasing proglottids in the stool. Those proglottids release eggs. The eggs contaminate grazing fields. The herbivore grazes on the field and ingests eggs. Those eggs become larvae in their muscles and encysted. And then when you eat the undercooked meat, you eat those cysts and infect yourself, and the cycle can start over again. Okay, then we have Tinea solium. This is the pork tapeworm. Now these worms are pretty long. They're usually around 5 meters long. Again, they're going to have a scolex with hooklets and suckers that attach to the intestine. Transmission is going to be through cycle C. So humans are going to be infected by eating animal meat that contains worm larvae. Distribution for this disease is worldwide. It's going to be concentrated in areas where humans live in close proximity with pigs or where they eat undercooked pork. So what happens is pigs are going to ingest the eggs. The eggs are going to hatch in the pig's small intestine. From there, the larvae are going to migrate throughout the organs, and they're going to encysted in the muscles. Now, cysticerci are the infective, encysted larvae that have settled in the muscles and the tissues of the animal. Teniasis occurs when we ingest these live cysticerci in the undercooked pork. The organism's coat is going to be digested, and then it's flushed into our intestine. There, it's going to attach firmly by the scolex and develop into an adult tapeworm, which then goes on to lay eggs in the intestine. Okay, so Tinea solium can cause a couple of different manifestations of disease. The first manifestation is going to be teniasis, and remember, this is when we ingest cysticerci, the larval stage that has encysted in the tissues of the animal. However, we can also ingest the eggs, and when we ingest the eggs instead of the cysticerci, another form of the infection occurs. What happens when we ingest the eggs is that we then become the host of the cysticerci. So cysticerci develop in us, and this is a condition called cysticercosis, one of the 5 neglected parasitic infections in the United States. So again, humans eat the eggs. The eggs are going to hatch in the human intestine and become larvae, and then the larvae are going to encysted in our muscles and organs. One of the organs that the larvae can encysted in is the brain, and when that occurs, a condition called neurocysticercosis happens. Epilepsy is going to be the most common presentation and complication of neurocysticercosis. Here is an MRI brain of a patient with neurocysticercosis, and you can see here the multiple cysts throughout the brain. And this can lead to neurological problems, including but not limited to epilepsy. All right, and now finally for this last part of the lecture, I just want to touch a little bit on the arthropod vectors of infectious disease. So arthropods, these guys have an exoskeleton and jointed legs. Arthropods are going to include insects like mosquitoes, fleas, and lice, arachnids such as ticks and mites, and crustaceans. Many of these guys are going to feed on blood and tissue fluid of us during their life cycle. These guys are called ectoparasites because they're parasites that live outside of the host. Okay, and then you can have biological vectors or you can have mechanical vectors. Biological vectors. These guys are going to transmit the infectious microbes in the process usually of feeding, but they also take part in the microbe's life cycle. Contrast this to mechanical vectors. These guys do not take part in any part of the microbe's life cycle. Instead, they simply carry the microbes on their body parts and transmit them that way. Okay, mosquitoes. These guys require an aquatic habitat. This is because their breeding sites are in water. Females take a blood meal, and that's how they're going to transmit disease. They transmit malaria, filariasis, dengue fever, Zika virus disease, and several other diseases. Fleas, these guys are highly motile. They have flattened bodies, and they feed on warm-blooded animals. These guys carry zoonotic diseases such as plague and murine typhus. Lice, these guys have small, soft bodies. They're going to attach to the head and other areas with body hair and feed inconspicuously on blood and tissue fluid. They release feces that contaminate the wound and cause diseases such as epidemic typhus and relapsing fever. So here's an image of a mosquito feeding on a human. On the right, we have a zoomed in image of a flea. You can see blood in its gut here after a blood meal. And this is what a louse looks like. And then we have ticks. Ticks are arachnids. These guys are going to cling on vegetation and then attach to the host when we make contact with them. Larvae, nymph, and adults are all going to get their blood meal by piercing the skin of the host. You have hard ticks and soft ticks. Hard ticks have small, compact, rigid bodies. They transmit bacterial, rickettsial, and viral diseases. Soft ticks, on the other hand, have flexible outer bodies and transmit relapsing fever. This is an example of the hard tick. Several different stages here, several different feeding stages, and you can see stages of engorgement. This is what the soft ticks look like.